whether there's glucose present, right? So when you mutate the operator for the repressor, that one of those questions drops out. Like we no longer have any information about, nor do we care about lactose, right? Because normally that repressor is what's asking or answering the question of what's happening with lactose. Okay, so now you've simplified the lac operon. Now it only goes as a result of what? Glucose. All right, and so it's only now responsive to what glucose levels are. So why is it that it's A and D and not B and C? Why is it that we see transcription with A and D?
Because it needs the activator to like push it along, and when glucose levels are low, cyclic AMP levels are high, so it's gonna push the inducer to say, let's go.
Let's go. Spot on. I'm not even gonna say anything. Okay? Questions for Allie?
So, like with this thing, so when glucose levels are really low, so that's why with A and D, when the glucose is absent, the cyclic AMP levels are high. And then you need that cyclic AMP because that's a co-inducer with the CAP, so then that attaches and then it pushes into.
Okay? So if you want more practice tonight, or when you go to tutoring tonight, actually, so I haven't said this, I think, in class. There is tutoring for this class, and it's Wednesday nights. So if you want to hang out with a tutor, Wednesday night, Caitlin, right? Caitlin, what's Caitlin's like? Fallon, that's right. Caitlin is the tutor. She's great. She and I meet on Wednesday afternoon anyway to her get up to speed. So anyways, so you can hang out with Caitlin, or you can hang out with your best friend from class, or just anyone who will listen to you. You should do the flip side of this. What happens if you mutated the operator for the CAP protein? Or what if I mutated both of them, right? Those are all valid questions, and you should be able to come up with answers to all three of those scenarios. Okay, so this is the type of thing that I do on my exams, right? Is that I'm never impressed with a monkey that's well trained, right? Which is what you are when you learn and memorize stuff, right? I will give questions like that because I think it's important that you learn definitions. I'm more impressed with your ability to think, right? So this is what I'll do. I will play God and make mutations, and then ask you to make predictions about what would happen. Okay? Levi.
So the operator deals with lactose levels?
Which operator are you referring to? The answer is yes, but make sure that we're talking about the right thing. Which of the operators deals with lactose levels? LAC-I. So the repressor protein. Okay? The CAP protein is dealing with and responding to glucose levels. Okay? So let's real quick do the second one. What if I mess up both operators? What's the consequence? No transcription, ever. Yeah, see if you can make sense of that. Why, Riley? Why is there no transcription?
Because there would never be that affinity created between the polymerase and the promoter by the cyclic AMP and the CAP protein.
Yep, spot on. Good job. Okay? This is the type of thinking that I'm expecting out of you. Okay? So, yeah, Bailey.
So is this case scenario, is that like lactose intolerant?
Yeah, so that's a good question. So, yeah, no, but here's why, right? So this is talking about who is this? Where is this lac operon found in? It's a bacteria. Right, so this is related but different, right? So the lactose intolerance that we deal with as mammals has to do with an enzyme, but it doesn't have to do with the lac operon. Yeah. Okay, so so basically what these next three slides are a host of different things that could go wrong. And so I'm just going to let this go for now, and I will leave this for Wednesday if you want to come back to them. But these are good places to start some conversations with people outside of class as you're studying. So you can see what this is saying is that this is I+, this is the LAC-I, you have a functional repressor, this is the wild type. What happens if you have a I-, a non-functional repressor where the repressor can't bind, or where you have a super repressor where it can't bind lactose? So these are all different scenarios. Like if you have godlike properties like I do when I write exams, I could come up with any of these scenarios and you should be able to come up with the rationale of what happened, right? So now what you can do is you can go through all the different components of this system and start tweaking them in all different ways and start practicing what happens, right? So this is one way that you can practice the type of thinking that I want you to get to, okay? So I'm gonna leave those for now, I'll let you play with them, and then we can talk more on Wednesday of next week if you want.
Can you explain the cis and trans acting? Like I understand the
Yep.
cis and trans like structures, but how does that relate to like the?
Right. So it's talking about the action of the mutation, whether it acts upon the operon itself or whether it has downstream effects. So like you can see like a super repressor, it can't bind lactose. This gets to levels of complexity that I think maybe are beyond where we need to be. But if you had a super repressor that can't bind lactose, you will never get it off. It will always be there, right? As opposed to if you had other mutations that would always, so if you had a mutation of the activator that would now always bound, even in the absence, that would always induce transcription. So this one is always shutting it down. There's other ones that will always turn it on. So the cis trans is asking what's the effect of the mutation and how is it positive or negative on the scenario that you're looking at. So for the most part, don't worry about trans or cis. I want you to understand the implications of what these things then do in the system. Okay? All right, practice them, see what you think. It'll be good exercises for your head, your brain. All right, so let's flip to this. This is the translation piece. Okay, so where we've come from, right? Way back when this class was brand new, we did transcription and talked about DNA structure, so way back over here. Right? Last week or the week before, we started talking about RNA polymerase and how transcription occurs. Right? So this is leading lagging strands, Ozaki fragments, RNA primase, all that sort of stuff. Then we started talking about RNA polymerase and how it works on DNA to do transcription 5' to 3'. And we talked a little bit about the different modifications of RNAs. Then we are now moving into, okay, now that we're here, that we have a message, and a message RNA, how do we process that thing and actually get it out of this is looking at the nuclear membrane, how do we get it out of the nuclear membrane and actually begin this process of translation? Okay? So this is the last step within this sort of funnel of information from DNA all the way to protein. All right, so you're going to see that we're going to focus on bacteria again, but we're going to be mixed and matching between prokaryotes, eukaryotes, because the big difference here is that prokaryotes, they don't process their RNAs, right? And the transcription and translation are conjoined processes. Eukaryotes, that's different, right? We process our mRNAs inside the cell, they come out of the, inside the nucleus, they come out and be translated in the cytoplasm. So just make sure that you keep those two things separate, right? That we're talking about two different processes that act differently because of the structure of the cells. Right?
Are we skipping the controlling gene expression slides?
Where's that?
The goals are, they talk about like E. coli and eukaryotic gene
Are we
regulation.
previous? Are we back here?
It's like separate from those slides.
Oh.
It goes in Moodle. We
Oh,
can
did it?
skip it. I'm good with that. But I just was making sure.
I could be skipping stuff. Let me see if we actually.
If we don't have time for it, it's okay.
No, well, let me just make sure that we're not. I think that probably. Yeah, well, let me see what I'm saying. I'm gonna skip before we skip it, but I would say yes. Are there any of these?
It talks more about like the lac operon and.
Let's skip them. You know what you need to know about the lac operon for this point. So, garbage. Oh, that's the wrong one.
What's example?
Yeah, in the wrong course too. It's like all sorts of struggle.
It's transcription control.
This guy?
Yeah.
oh, this is where all the lac stuff is. Yeah, so,
So you
that-
just skipped it and explained it.
right,
separately,
yeah,
right?
I just drew it on the board,
Okay.
yeah. So, I won't take, I mean, the content, so the transcription factor stuff is dead. Don't worry about any of the stuff at the end, but all this lac operon stuff, I was wondering where all those slides went. I'm not sure why I don't have them together. So this is what I drew on the board for the last couple days, because I was like, where are all of my lac operon stuff? But it's all right here. It is. Cool. Nice. That would have been helpful to put up for you guys. I apologize. But yeah, so lac operon is live. That's good. And this is all that we just went over. All that. But after that, the control of gene expression in eukaryotes, you can forget that. And transcription factors, you can forget that. Okay. Thank you, Riley, for bringing that up. Okay, so back to this. So we're going to look at the process of how that we do translation. We're going to talk about the codon, the generacy of the code, which you've seen before. And we'll talk about the tRNA, mRNA, how that they work. I'm gonna come back to this. These are terms that I'm gonna use. We could break up and do these definitions, but I'm just gonna go forward, assuming that you have some knowledge of this already, and if there are things that are confusing, just stop me and we can do that. But we'll talk about this in the relationship as we kind of go forward. Okay. So we mentioned this briefly. I want to give you a little bit more in depth of how this happens. So we're talking about, this is now we have an mRNA transcript, and the context is that we're talking about a eukaryote, okay? Because we're talking about processing the mRNA, and that only happens in eukaryotes, okay? So what you see here is the following, is that somehow that you're going to have to remove introns and splice exons together. And so this is showing you one of the ways that processing happens. So what you're looking at here, this is called a self-splicing mechanism. So you can see that the mRNA itself splices out the introns without any additional protein machinery necessary to do that work. Okay? So there is examples where protein machinery will help do that splicing work, but this is showing you a self-splicing one. So here's the scenario. Here's your DNA, and in that DNA there's a gene, right? And there would be a promoter here, and the RNA polymerase will bind to that and make a primary transcript. Okay, so we say primary transcript because it's the unprocessed straight RNA from the DNA code, but you need to process it in order to become a mature mRNA. Okay, so what we're trying to do is that these pink things here, we need to get rid of and just let all the exons be next to one another. Okay? So that's our goal, is to get rid of the introns, ligate the exons. Okay? So notice that we talked about this before too, is that remember the 3' N, that there's a poly A signal, that the poly A polymerase binds to that, cuts off the N, and then polyadenylates the tail, right? That this is from 2 slides before. This is one of the processing steps, so that's showing you that here. And it's also showing you that there's a 5' methylated guanine. So this is the 5' cap that you're seeing. Okay, so primary transcript has already got its cap on and its tail, and now we just need to get these exons together and introns out. Okay, you with me so far? Okay, so this is showing you the splice that's happening. So this guy here goes through this sort of looped structure, and we'll talk more about this, but these splice sites, their specific sequences, don't really care that you know them, but these splice sites will help to determine where the boundaries are for the introns. So it will make this what's called a lariat structure, a lariat like a lasso of calf sort of thing. So a little lariat structure that will literally splice itself out in both scenarios, that will then join the exons together. Okay.
Where do they go?
Where do they go? So, where is this happening? Where is all of this occurring? In the nucleus. So this is now RNA that can now be recycled. So all these bases will be chewed back up again, all the way down to their bases, their A's, T's, U's, and G's. A, U, G's, and C's, and reused in the next process. Okay. Once you have all these together, this now is the mature mRNA that can be exported out of the nucleus into the cytoplasm. Okay? So 3 things needed to happen. Cap, tail, exon. All 3 of those are the processing steps for the mRNA. Okay. Here's a more in-depth view of what this looks like. So what you see is that so this is zoomed in, so exon, intron, exon, 5' splice site. They always have this is the consensus sequence. Remember we talked about consensus for promoters? This is the consensus splice sequence. It always goes A or C, AG, GU, A or G, AGU. Okay, so what you're seeing is that this is, if you're looking in a mRNA transcript, this is what you're looking for as far as define where these splice sites exist. And that will determine the boundaries of where that will happen. Okay, so I'm not expecting that you memorize consensus sequence. I want you to just see that there are boundaries that we know, and that this is what is determining that lariat formation. Okay, so so this is showing you the process now zoomed in. So the lariat is formed by this first exon swinging over, this 3' OH that exists on the end of that one exon comes in and makes a join with the exon 2, and that's how that splice ends up happening. Okay, so I'm not going to ask details of that. What I want you to understand is that with these eukaryotic messages, there has to be a mechanism that allows these introns to be spliced out. This is showing you what's called self-splicing. Like all that is needed is just the mRNA. Okay. There are other examples where proteins will mediate this work for you. Okay. Similarities, just make connections in your brain. Transcription termination happen independent of any protein factors. The sequence itself determined transcription termination. That's true for here as well. The sequence itself of the mRNA was sufficient to result in splicing. Okay? You can imagine there's other scenarios where protein machines will do this work, but in this case, all that you need is just the transcript. Okay? All right.
Why does it want to get rid of the introns?
Yeah, so so part of this is that we like to make clouds look like bunnies and somehow have this scenario that somehow these things are thinking about, why would I do this? This is just driven by chemistry, right? That these reactions will occur because of, and not always they will occur at some frequency, but this, you can see it makes these base pairs, and then that then drives the chemistry of that this 3' can actually do this, right? So why does it do it? Because the reactions are favorable for making that to happen, right? So if these things were sensitive, that they actually thought about this, the rationale from our end is that this needs to happen in order for a fully processed DNA or mRNA to be made, so that you actually have a functional DNA or a functional protein, I guess. But this is all driven just by favorability of reactions. And I guess the other thing I should say is that if it didn't have this combination of sequences that you see here with this polypyrimidine track, that stuff wouldn't happen. So if you mutate some of these things here, what will end up happening is that you're not going to have that splicing event occur. And likely what will happen is that now you'll have an intron that's left in the mature transcript, and that will have consequences as far as what sort of protein that you're actually making. Okay, so thinking about applications of this, if in my godlike powers I begin to mutate some of these consensus sequences, introns don't splice out. They don't splice out, now you have a very mutant protein, right? Because you'll have this huge intron that's now part of a protein that really shouldn't be there. Levi?
What's the point of having introns if they don't go through?
Yeah, and so this gets at the bacteria. Like bacteria don't do any of this, right? They just straight have the code, and that's what the code is. So we don't know. Like it seems to be that so the one positive is that with eukaryotes you can do the alternate splicing thing, right? So you have some genetic efficiency where you could make 4, 5, 6 different proteins from the same combination of exons by just mixing, mashing the exons together. So that's one, but we don't know. Like, there's not a good answer for that. All right.
Does this relate to, there was a science event last semester about, I think it was about like mosquitoes or something like that, and how they were trying to like mutate their genes, and they showed like the intron exon like
Did they? I don't know. I didn't.
Yeah, I remember that.
Yeah.
Was it alternate splicing stuff?
Yeah, I think it was like changing.
Was it related to this?
I think so.
Okay.
Just a question.
Cool. I don't know. Sorry.
You should go to more science events.
I totally should. Wish someone required me to go to those. That'd be good. All right. So what this shows you is the same splice process now done with proteins. Right. So right. So all you see here is that now you, instead of having a self-splicing like you did before, now you have these proteins in blue and green. This is protein U1, U2, and that when U1 and U2 come together, that there's another protein in here that helps physically cleave off these different introns and splice the exons together. So very similar to the mismatch repair, you have these two proteins that are supposed to look at these different sites, and when they come together, then at that point, the protein now has exonuclease activity and can cut, right? But when they're separate, they don't have that activity. When brought together, they cut and then paste things back together. Okay, so this is the other scenario, right? If it doesn't do it itself, then there's machines protein machines that will recognize and do that for it. Okay? As you might expect, these recognition sequences are different than the one that we previously saw. So some transcripts will go through this mechanism, some will go through the previous. And what will matter is what these sequences are and whether this is the preferred method or this is the preferred method. Yeah, right.
So that protein that took the intron down, like over U2
Yep.
at the bottom,
Here.
right there. So does that just unbind and then it just breaks apart, or is there something else that'll break it apart?
Yep, same thing. So once the exonuclease activity goes, that releases, the protein will release the RNA. The RNA will be chewed up, recycled, and then those proteins go back again, look for more sites that they can bind to. Okay. All right, so where we've come from now. So we have the cat, we have the tail, we have a spliced together exons. Now what has to happen is it has to get out. Okay, so you can see there's a couple different scenarios that happen. So why did we go through this? What's the point? So remember that what needs to happen now is that, well, one of the things that needs to happen is when you're in the nucleus, you want to make sure that only messenger RNAs that have been fully processed get themselves out of the nucleus, right? So if you have a primary transcript that has a 5' cap but doesn't have a tail or hasn't been processed yet, that the cell needs to figure out ways to make sure that it doesn't accidentally push mRNAs out into the cytoplasm until they are fully acted upon, okay? So what this is showing you is that there's ways for that to happen, and the biggest is that the nuclear pore complex, right? This is a host of proteins that exist in the nuclear envelope. These nuclear pore complexes are basically checking for the requirements that are out there, right? So what you can see is that there's a protein that is recognizing exon junction complexes. There's a protein that's looking for a cap, and there's a protein that's looking for the poly A. Those are the three things that we just said that the mRNA needs to have done to it before it's actually fully processed. This nuclear pore complex is looking for those 3 check marks, which is exactly what you would hope would happen, right? Is that, did someone put a cap on? Did someone put a tail on? Did someone make sure the introns are out, right? If those 3 boxes have been checked, we approve this message and allow it to go out to the cytoplasm, okay? So that's what's happening here. Then once this thing is out there, then these things can come off, because now this message is good to be translated.
Are we gonna learn how those bind on there, like how, like the protein?
Not other than the fact that it's going to bind, so this poly A binding protein, its recognition sequence should be A. So, right, so this thing is going to look for that methyl, that 5' methyl of that guanine, right? So they're looking, and these exon junction complexes, it's a little more complicated, but they're looking for those sequences that would exist if that has happened. Other than that, no, we're not going to go to the depths of the sequences of what's happened there. Okay?
So the nuclear core complex is recognizing those proteins?
Yes. So the core complex acts as a door or as a gate, right? The gate remains closed until all three of those proteins have bound to that complex. The complex then changes its shape, allows the message out, and then comes back again and shuts. Okay. Okay. All right, so are we good? Straightforward? I paused, but I don't think there's anything there's a question here. It's just, this is what it goes on. All right, so let's begin down. So we'll go to the places, if you've already seen lots of the beginning parts, we will pick up on Friday with the places that you probably haven't seen. So I expect that you already know this. So, right, you know the basics of translation, true? Maybe? No? All right, I don't know what any of that means. Okay, so here we go. Translation, right? So we're at a place where the messenger RNA is now out in the cytoplasm, and how do we get that message to make a protein? All right, so if you're not aware, there are 64 different possibilities if you have 3 nucleotides. Like if you do all the combinations of what those 3s could be, 64 different codes, okay? There's only about 20 common amino acids. There's a couple of different other ones, but 20 common amino acids. So what that means is that the codon chart is what's called degenerate. That means that there will be several codons for every amino acid, right? So that's the degenerate part, okay? And this is really the kind of cool thing, is the code is universal across all of life, from bacteria to plants to humans to anything in between. We all use the same code, which is kind of crazy. But this is the one system that remains constant throughout, okay? And the other similar codes encode similar amino acids. So if when you get to biochem, we'll learn more about the amino acids, but amino acids have specific properties. Some of them are nonpolar, some of them are polar, some of them positive, some of them are negative. So those that have similar properties have similar codes with them. There's a grouping of them together. So if you haven't seen, this is what a codon chart looks like. And so what this is showing you is that the first letter of the codon is on the left-hand side, the second letter is on the top, and the third letter is on the right. So this valine right here, the codon would be G, U, G. Okay. So you can see that there's a whole host, right? So here's your valine, your alanines, your threonines, your prolines, all sorts of different groups, clumps of the 3 codon letter and how that relates to the amino acid that exists. Okay. What you see over here are codon and anticodon. So this is the tRNA that we talked about before. tRNAs are never translated. All that they do is do this, right? So tRNAs have an anticodon loop, and then on the very top of them, they have an amino acid attached to them. Okay. So this histidine, you can see that histidine is C, A, C. So notice that the anticodon loop is G, U, G. Okay? So this is the basics of how translation works, right? The codon, anticodon will match perfectly with their base pairing, right? And that they will put in the appropriate amino acid to the growing polypeptide chain. Okay? All right, so done talking about questions on this? This should be all straightforward from previous classes, I would think. Okay, the other part of this is that the code, so there's lots of ways this could have happened. The code also is non-overlapping, which means that you don't, so if the code is, right, it comes in sets of 3 like this, and not like like, so that'd be one possibility, which is what it is. It's not this, not ATC, TC code, right? So here's our big red dog bit the old man, and our 3-digit code, 3 letters all the way through. So notice that if you put in a random, this is called an insertion, right? So you had an insertion deletion, and notice that what happens to the code is that every point after that insertion is now screwed up, right? So now your code doesn't make any sense after the X being added in. So R, X, B, G, the whole protein is screwed. Okay? So insertion and deletion mutations have, from that point on, have domino effects that the protein is now completely off. So those are big deals. Okay, so those are those. Both of these would be what we would call frame shift mutations, because it has shifted the frame out of the one that made sense to read. Right? We used to be able to say something that made sense. Now the frame is shifted, so none of this is all garbly poop. So every amino acid after that mutation has now been changed. These, what you can see happens here, when now that you're just playing with scenarios, right? So what happens if you had an insertion mutation and then you had a deletion after that? Notice that deletion restores the frame. So you had a frame shift, but then you went back to the old frame, right? So, and this you could do all sorts of things, right? What if you had 3 insertions all in a row? You would eventually restore the frame, right? Because you shifted it once, you shifted it twice, you shifted it 3 times. Now you're back to the same frame you were before. Okay? So they're just showing you all the different ways that mutations can occur and the effects of those mutations. Yeah, right.
So for these mutations versus like the mutation that you did on the operator in transcription. So for like the operator in transcription, when you say like that's mutated so it's not there, is that like a sequence that's gone? And this is not a sequence, this is just a single like spacing between letters?
Right. Yeah. So so when I screw up stuff in the operator, it could be lots of things, but either I could take it out completely, but the operator isn't actually part of the actual transcript, right? This is part of the promoter agent. It's not part of the thing that's being transcribed. And this is different, right? This is talking about message when the message itself has been changed. So in the actual transcript, you have things that have been changed, and not just in the transcript, but actually in the coding region of the transcript. So not in the 5' UTR, not in the 3' UTR, but in the actual coding region. Mutations there are critical. The other thing is that not shown here, but these are talking about insertions and deletions, but that doesn't mean, so like the mismatch repair that we talked about, that's not an insertion or deletion, that's just a change, right? That you went from G to C. So that would be a case where you said, our Bill red dog bit the old man, right? So those are not frame shift mutations, they just changed the one letter. So one word may be off rather than the whole sentence. That primarily is what happens, is that we have a change in base, not a deletion or insertion of base. So this is why deletions and insertions are big deals. They screw up everything, not just one thing. Okay, so this is where we'll start next time. We'll talk about the tRNA, and we'll also talk about the process of actually doing the elongation of translation. And we'll see where we're at from there. Okay? Have a great afternoon.